my name is Debbie Sunday, and I'll be discussing the light-activated natural product extracts for photodynamic inactivation of bacteria. First, photodynamic inactivation, also known as PDI, is a light-triggered antimicrobial strategy that is used to target and destroy abnormal cells. This treatment utilizes a photosensitizer that can be activated by specific wavelengths of light to generate cytotoxic singlet oxygen and other reactive molecular species, RMS, for destroying unwanted and highly resistant bacterial pathogens. The reason natural product extracts are being used in this study is that many naturally occurring compounds in plants are produced as secondary metabolites, and they function as photosensitizers and complex defense mechanisms against herbivores and pathogens. Photosensitizers can be further classified based on their structure and chemical origin. Some examples of natural product photosensitizers include anthraquinones, benzopyrenones, and flavin derivatives, whereas synthetically derived photosensitizers include porphyrins and phenothiazines. Antimicrobial resistance presents a problem with a clear unmet need, and our objective to counter this problem is to develop photosensitizers that can produce an immediate burst of relatively nonspecific cytotoxic singlet oxygen in order to prevent the bacteria from gaining enough time to gain resistance. Other RMS, along with the cytotoxic singlet oxygen, in this process can overcome antimicrobial resistance that can selectively target highly resistant bacterial cells. These cytotoxic singlet oxygen and RMS species can be produced through two mechanisms. One mechanism is type 1 electron transfer, where the photosensitizer absorbs light and becomes excited, and transfers its energy to nearby molecules, which can then react with other molecules in the cell and leading to cell death. Type 2 energy transfer involves the activated photosensitizer transferring its energy directly to atmospheric oxygen in its triplet form. And this then leads to the formation of singlet oxygen molecules that are cytotoxic and can damage the cell membrane, DNA, proteins, and ultimately lead to bacterial death. The natural product extracts used in this study include Japanese knotweed, turmeric root, aloe vera, rhubarb root, and yellow dock. While most of these natural products were selected at random, some of them, such as Japanese knotweed, were selected with the pre-existing knowledge that they contain photosensitizers. The workflow of this overall process starts with the extraction of the natural product from its original environment. The natural product is then soaked at room temperature and with ethanol. Ethanol is our choice of solvent because while it is easy to use, inexpensive, and accessible, it also disrupts the plant cell membranes. Following the room temperature soak, the natural product extract is then filtered and the solvent evaporated. To optimize our percent recovery from the original product, the natural product is extracted either by soxhlet, which is a continuous cycle of solvent evaporation and condensation, or by reflux, where the reaction mixture is heated to boiling point, vapors are condensed, and then returned. The antimicrobial properties of our extracts were then studied further in an agar diffusion assay, where various bacterial strains were inoculated on an agar plate. Wells were made using sterile pipette tips. The natural product extract solutions, which were then made in DMSO, were then pipetted into the wells, 20 microliters each, and each of the plates underwent light treatment. The plates, or the experiment rather, was replicated in both dark and light conditions. After 20 to 24 hours of incubation, the zones of inhibitions of each of the products or extracts were observed and recorded. In these results, Japanese knotweed is shown in both dark and light conditions, and clear zones of inhibitions can be shown in the light conditions across the various bacterial strains. The table above also shows the zones of inhibitions of all the natural product extracts in their various forms across several different bacterial strains, allowing us to observe the ones that are firstly active in the light and those that may be more active than others. These results will be validated using more quantitative approaches, such as minimum inhibitory concentration assays, as well as bioassay guided fractionation to optimize the photocytotoxicities against the antimicrobial resistant strains for the lead natural product extracts. We will then be able to compare their antimicrobial and photoantimicrobial properties against other bacterial species as well. We thank the Department of Chemistry and Biochem at UT Arlington and also the National Science Foundation grant. Thank you.